Okay, so welcome to this next video in which we are discussing the mammalian circadian clock. Okay, so we're now discussing how these axons coming from these photoreceptive ganglion cells within the retina are going to influence the neurons of the suprachiasmatic nucleus. Now, we've discussed that they will release two neurotransmitters onto the neurons of the suprachiasmatic nucleus, which are glutamate, the main excitatory neurotransmitter of the brain, and also PACAP, standing for pituitary adenylate cyclase activating protein. What we now want to discuss is how glutamate, acting on receptors on the surface of the suprachiasmatic nuclei, neurons, is going to trigger a shift in the circadian clock, basically, within the suprachiasmatic nucleus neuron. Okay, so if the circadian clock is out of phase with the light-dark cycle within the suprachiasmatic nuclei neuron, you're going to get the circadian clock reset, basically, by the light. Okay, and we want to see the pathways that underlie this. So, the suprachiasmatic nuclei neurons have on their surface ionotropic glutamate receptors, okay, which are the main forms of glutamate receptors within the human brain. Okay, so ionotropic glutamate receptors are ligand-gated ion channels, okay, and they're often for short abbreviated to the IGLURs. Now, there are three main families of ionotropic glutamate receptors, which are the Ampa receptors, the kainate receptors, and the NMDA receptors. However, we're not going to initially distinguish between them because for the first part of our discussion, which is the basic structure of these receptors, they all have the same basic structure. Okay, they're all tetramers and the subunits all have the same basic structure. So we're going to look at that first, which is general for all of the different types of ionotropic glutamate receptors, and then we'll focus in on the three different types of ionotropic glutamate receptors and see their involvement in the resetting of the circadian clock. Okay, right. So we're going to start off then with the basic structure of an ionotropic glutamate receptor. So, let these two lines represent the inner leaflet of the phospholipid bilayer and the outer leaflet of the phospholipid bilayer. So I'm drawing the cell membrane, okay? And we want to draw an ionotropic glutamate receptor. Now, ionotropic glutamate receptors are ligand-gated ion channels. Whenever drawing a ligand-gated ion channel, it's a good idea to start by drawing a cylinder with a hole down the middle through which the ions can move. Now, ligand-gated ion channels usually have a very low open probability before the ligand is bound. So the chance that they will be open when the ligand is not bound is very, very negligible. Okay, but once the ligand is bound, their open probability is going to increase significantly. Okay, so what's the structure then of these ionotropic glutamate receptors? Well, the entire ligand-gated ion channel is not a single protein. Instead, it is a tetramer. It's made up of 4 separate polypeptides, which are all joined together to make the full ligand-gated ion channel. Okay, so it's made up of 4 quarters, essentially. What we now want to do is we want to pull out one of the quarters of this ionotropic glutamate receptor. So I'm now highlighting in purple one of the subunits that makes up a quarter of this ionotropic glutamate receptor, and I want to pull this subunit out and have a look at its structure. So, again, it's going to be a membrane-spanning protein, so I will draw the membrane here. Okay, and I'm just a little bit concerned that I haven't left enough space, which I haven't. Okay, we'll draw it again down here. We've got more space. So this is the outside of the membrane, the extracellular fluid is on this side, and this is the cytoplasmic side of the membrane down here. Okay, right. So the individual subunit that makes up a quarter of an ionotropic glutamate receptor is a single polypeptide. Okay, so it will have an amino terminus, which is extracellularly, and its carboxylic acid terminus will then be intracellularly. Now, I'm then going to draw a line to represent a polymer of amino acids. So this little line that I've drawn here, this is amino acid after amino acid after amino acid after amino acid, etc. Okay, then you come across an important domain, okay, which I'm going to box here. So this box, this represents a domain that's often called the ATD. Now the ATD stands for the amino terminal domain. Okay, so it's called that because it's near the amino terminus of the protein. Now the amino terminal domains of subunits of ionotropic glutamate receptors are important in the binding of the 4 subunits of the ionotropic glutamate receptors together. Basically, it's important in the binding together of the separate subunits. Okay, right. After that, what will happen is the polypeptide is going to fold around in a way that I will draw like so. Okay, and this portion is going to be very important in forming the ligand binding domain, but we'll come back to that in a moment. Okay, then you have your first membrane spanning alpha helix. So the polypeptide is going to span the membrane. It's going to go from being on the outside of the membrane to being on the inside of the membrane. Okay, and although I've drawn this as a straight line, in actual fact it will be an alpha helix, and therefore its structure will be more spring-like, like so. So it will have a structure like this. Okay, and that will straddle the membrane. It will have a bunch of hydrophobic residues within this alpha helix here, and these will integrate nicely into the phospholipid bilayer. And then what will happen is the polypeptide will try to get out of the cell again, so it will try to pass through the membrane again, but it will fail and it will come back. Okay, and when you have a structure like this, where the polypeptide tries to get out of the cell and then comes back in, it's known as a re-entrant loop. Okay, so there's our re-entrant loop. Then we have our second full membrane spanning alpha helix. Okay, the polypeptide then remains outside for a portion, loops round, forms an important part of the ligand binding domain. As I say, we'll come back to that in a moment, and then is going to recross the phospholipid bilayer for another time. Okay, and then it will end with its carboxylic acid terminus in the intracellular compartment. Okay, right, so let's highlight up different important portions of this structure. So we'll highlight the amino terminal domain in turquoise here. Okay, and as I said, that's important in binding the subunits that make up the full ligand-gated ion channel together. Okay, then the next bit I want to highlight is this portion here. Okay, this portion is still in the portion prior to the first membrane spanning alpha helix. Okay, and this special domain that I've highlighted in blue is known as the S1 domain. Okay, then continuing on, I'll come back to the S1 domain. Okay, let's talk about the membrane portions. Okay, so we have 3 membrane spanning alpha helices, and we also have this re-entrant loop. Okay, and these are the membranous portions. These are the portions that are in the membrane. The first membrane spanning alpha helix is called the M1 domain. This re-entrant loop is then called the M2 domain. The second full membrane spanning alpha helix here is known as M3. Okay, and then the final membrane spanning alpha helix here, which is actually the third membrane spanning alpha helix, but which is the 4th membrane domain, is known as M4. Okay, right, so you have these 3 domains which are within the membrane, labelled M1, M2, M3, and M4. Okay, right. Finally, the last important domain I want to discuss is in this loop between M3 and M4. So this loop in between M3 and M4 is known as the M3-M4 loop. And we have a special domain here that I've now highlighted in green, and this is known as the S2 domain. Okay, and together the S1 domain and the S2 domain are going to form what's known as the LBD. Okay, which stands for the ligand binding domain. The L is for ligand, the B is for binding, and the D is for domain. Okay, so the S1 domain of the pre-M1 region and the S2 domain of the M3-M4 loop together make the top and the bottom, if you like, of this ligand binding domain. So basically the ligand is going to come and bind in here. Now let's just discuss the ligand for a moment. What is glutamate? Okay, glutamate is a small molecule. It's also an amino acid. But I would just like to discuss what the difference between glutamic acid and glutamate is. So let me just firstly draw you the structure of glutamic acid. Okay, so it's an amino acid. So let's start by drawing the core amino acid structure. So here's the amino group. Here's the alpha carbon with a hydrogen coming off it. Here's the carboxylic acid group. Okay, like so. So that's just the core amino acid structure. All amino acids have that. Okay, all proteinagic amino acids anyway. And then the R group of glutamic acid is a three-membered carboxylic acid group. Okay, so we have two methylene groups like so. And then off the side of this, we then have a carboxylic acid group. Okay, and this is the R group of glutamic acid. So this is the structure of the glutamic acid amino acid. Okay, now, what then is the difference between glutamic acid and glutamate? Well, glutamic acid and glutamate, the difference between them is that glutamate is the conjugate base of glutamic acid. Now, what does that mean? Well, basically, the reason glutamic acid is called glutamic acid, okay, stressing the acid here, is that it is indeed an acid. It has an acidic R group. Now, I'm not talking about this carboxylic acid group here. All amino acids have that. Ignore that. What we're interested in is this carboxylic acid group that you have within the R group here. Okay, that is capable of donating away a proton into the solution. Okay, so carboxylic acid groups are acidic because they are capable of donating protons away into solution. That's the characteristic that you need to have in order to be considered an acid. So what can happen basically is that if we draw this bond larger, here's the oxygen with a hydrogen attached to it. It's a covalent bond, so there's one electron coming from the oxygen here and one from the hydrogen. Okay, so let's say this one came from the hydrogen, this one came from the oxygen. What can happen is that the hydrogen can fall off and leave its electron attached to the oxygen. Now, hydrogen only consists of a proton with an electron orbiting around it, at least the normal radioactive, the normal isotope of hydrogen only consists of a single proton in the center there in red, with an electron orbiting around it here in blue. Okay, so if it leaves its only electron behind, then you're just giving away a proton basically, and this proton will then go into solution. So basically, glutamic acid molecules can lose that proton off the oxygen, leaving the oxygen with a negative charge, and that's why they are an acid, because they can donate protons away into solution. Glutamate is then the molecule that is left over after you've donated the proton away. So after you've donated away the proton, you've got this oxygen with a negative charge here. That is no longer an acidic group there. It can't donate away a proton again. Instead, it's actually a basic group. Okay, because that oxygen with the negative charge can accept a proton onto it, okay, and therefore it satisfies the definition of a base, which is just a molecule which can accept a proton from solution. Okay, so this is known as the conjugate base then of glutamic acid, and this is a running theme for all acid molecules, that once they donate their proton away, the molecule that is left over is no longer an acid. Instead, it's a base, and it's called the conjugate base of the acid. So glutamate is just the conjugate base of glutamic acid. Okay, now, the reason everyone talks about glutamate rather than glutamic acid is that under physiological pH, most glutamic acid molecules have actually given away their proton to solution and therefore exist in the glutamate form. Okay, so they're sitting in the glutamate form rather than in the glutamic acid form. Okay, so most of these sort of molecules will have lost that proton and therefore will be actually glutamate molecules, and very few of them will be glutamic acid molecules. So that's why everyone obsesses over glutamate and never talks about glutamic acid. Okay, so that's the ligand, and basically it's going to bind in this domain formed by S1 and S2. Okay, so that's the ligand binding domain. So that means that every single one of our subunits of the overall ionotropic glutamate receptor has its own ligand binding domain. Okay, right. So just to remind you that the full ionotropic glutamate receptor then is formed by 4 of these all tetramerizing together, basically. So you'll have 4 separate ligand binding domains. What we now want to turn our attention back to is the 3 different families of ionotropic glutamate receptors. There are actually 4 different families of ionotropic glutamate receptors, but the final family, which is the delta glutamate receptors, is just not worth talking about. They are not receptors for glutamate. No one knows what they're receptors for. We have not found an endogenous agonist so far. We don't know what they're doing, basically. So we're not going to talk about those. We're going to talk about the ones that are better understood, which are the Ampa receptors. Okay, and again, there is not just one Ampa receptor. It's a whole family of receptors. There are many different types of Ampa receptors. So the ionotropic glutamate receptors is the big superfamily, if you like. And now we're subdividing them further. There are the Ampa receptors. There are then the kainate receptors. Okay. And by the way, the Ampa receptors for short are often abbreviated to the AMPAs. Okay, so let me show this. AMPA, AMPA, and then you put an R after it, so Rs, AMPAs. Kainate receptors are often abbreviated to KARs, which stands for kainic acid receptors. So again, kainate is the conjugate base of kainic acid, so they're very much so related. Okay, and both of these are named after agonists that we have found for them. So Ampa is some chemical which is an agonist for Ampa receptors. Kainate is a chemical which is an agonist for kainate receptors. Finally, the NMDA receptors. NMDA is an agonist for the NMDA receptors. Okay, so all of those chemicals bind to the ligand binding domains of the subunits of these receptors and activate them. Okay, and similarly, the NMDA receptors are often abbreviated to the NMDARs. Okay, right. So, in the next video, what we will start doing is looking through these 3 different families of ionotropic glutamate receptors. Okay, we'll start with the Ampa receptors. We'll look at the different subunits for Ampa receptors. We'll look at how they can tetramerize together, and we'll then look at the effect of glutamate on them and what the effect on the cell, the postsynaptic cell of these Ampa receptors opening is. We'll then move on to the kainate receptors, which shouldn't take long because they're almost identical to the Ampa receptors. Okay, so we'll discuss the different subunits of kainate receptors, and then we'll parallel what happens with the kainate receptors when they open to what happens with the Ampa receptors. Then we'll talk about the NMDA receptors, which are the most complicated, okay, and what happens when they open. Okay, right, and then we'll see how these receptors opening leads to a calcium signal, mainly we think through the NMDA receptors, okay, and how that calcium signal is then going to lead to the resetting of the internal circadian clock within these suprachiasmatic nuclei neurons. And then we'll turn our attention to PACAP, or PACAP.